Well, welcome everyone. It's my pleasure to welcome you to this webinar today on clonal hematopoiesis. My name is Todd Druley. I'm the Chief Medical Officer for Mission Bio, who's hosting this event. And it's my pleasure to moderate three really dynamic and exciting researchers in the area of clonal hematopoiesis. So it's my pleasure to introduce Ross Levine, who is the Director of Translational Research at Memorial Sloan Kettering. Kelly Bolton, who's an assistant professor of internal medicine specializing in hemato-oncology at Washington University in St. Louis, and Jyoti Nangalia, who is a cancer genetics and genomics researcher for Cancer Research UK and their clinical scientists and hematopathology consultant division. So I joined Mission Bio earlier this month after a long career in academic research myself, because I'm very excited about the future of single cell multi-omic research. And I think, clonal hematopoiesis as a subject and as a matter, we really only just scratching the surface of what it means physiologically and pathophysiologically. And so, it is a field that's really ripe for this type of research. And so this trio of investigators has done some of the most leading work in the area. And so with that said, I'm going to turn things over to Ross Levine, who's going to give us a short overview of where the field of clonal hematopoiesis stands. But before I do that, I want to make sure everyone realizes, if you have questions, please don't put them in the chat box. Please put them in the Q&A. There's a little tab down at the bottom of your screen if you move your cursor. So if you put your questions in the chat box, they're probably going to get missed. Please put those in the Q&A section. And later on in this webinar, we'll be sure to get to as many of those as we can possibly get to. So thank you very much. And with that said, I will turn it over to you, Ross.
Thanks so much, Todd. It's great to be with all of you virtually. Although, as I think some of you know, if you follow me on Twitter, I'm on day 5 of my COVID isolation, and I hope everyone else is doing well and safe wherever they are. So what I thought I would try to do is maybe set the stage a bit for what will be, I think, a great dialogue with two colleagues who I think will have amazing insights and ideas. And I can't say how excited I am to have Todd both here today at the helm and to see him join this great organization at Mission Bio. I'll just say for COI that I am a scientific advisory board member of Mission Bio, and so I just want to make that clear from the outset. So I just want to sort of level set a bit and maybe indulge you all in a little bit of history here. I'll remind you that this field of clonal hematopoiesis with respect to sort of genetics and genomics is new, but the concept is not. And really, if you go back to work done in the 70s by Fialco and colleagues using sort of X inactivation studies, and then subsequently work in the 90s and 2000s by a number of groups, including my mentor, Gary Gilvan, with one of his early postdocs, Lambert Busk, who's shown here, it was well known that non-random X inactivation in heterozygous females was an age-associated phenomenon, suggesting the possibility that there was stochastic clonality in the hematopoietic compartment with age. And yet, for most of the 90s and 2000s, there was a vigorous debate about whether clonal hematopoiesis was an artifact of the assay, a sort of random walk towards clonal, like the few stem cells left, was really being driven by cell intrinsic or extrinsic events. And it wasn't until a conversation that Lambert and I had at ASH at a hotel bar when we were discussing this phenomenon in the early days when exome sequencing was really being developed, that we said we could solve this once for all by doing some sequencing of women that had presumed clonal hematopoiesis based on X activation of their blood or epithelial cells. And we identified at the time, which was a surprising result, which were recurrent TET2 mutations in elderly women who had no blood cancer, no blood count abnormalities, and had easily detectable TET2 clones by Sanger sequencing. So they're at a very low frequency of 5, 10, 20, 30%. And the frequency in healthy individuals was about 3 to 5%, so about a third of the frequency in AML, and maybe about a fourth or a fifth frequency in MDS. And we actually had one informative case at the time that had a TET2 mutation, and then 8 years later developed essential thrombocytosis, and at that time acquired a JAK2 mutation and had the same TET2 mutation. So that provided proof that it had to be in a stem cell with self-renewal that could exist for 8 years and could acquire a secondary event that gave rise to disease. And if you then look at the sort of mechanistic work in parallel, a number of groups, including our own, as well as William von Schenker and Olivier Bernard and others in France, made conditional knockout models of TET2 based on its observation that it was a MDS and AML gene. And when you knock out TET2 in mouse models, you get an increase in the fitness of stem cells, but a very, very, very low penetrance of overt disease. And we can have an interesting conversation about whether these mice develop leukemia without extrinsic or additional intrinsic factors. But the point I want to leave you with is that in the human, TET2 increases stem cell fitness and drives clonal hematopoiesis. And in the mouse, you largely, when you inactivate TET2, recapitulate that phenomenon. So the preclinical and clinical data really do match up. And we now know that clonal hematopoiesis, and I like to use that term, other people like the term ARCH. You can use that term, especially if you're from St. Louis, like Todd, or you can use the term clonal hematopoiesis of indeterminate potential, or CHIP, as you can see the chips in the top right on my slide. Whatever you want to call it, we know that it's a pre-malignant clonal state induced by somatic mutations. And we know that this can occur both in patients who have overt disease, and you can find the antecedent cells. And if you do population-based studies, there's the really seminal work by Sid Jaiswal and Ben Ebert and Steve McCarroll, that in 10 to 15% of the aging population, you can find these mutations. The implications clinically, which we'll talk, I think, in detail about today, are expanding. And we're just understanding what this means, both with respect to leukemia risk, with respect to following people after leukemia treatment, and the risk of non-hematopoietic side effects. But I'm, as a clinician and scientist, the most excited that in the long term, I think this may be our first frontier for effective malignancy prevention, and that we may be able to identify the higher risk patients and intervene, but we need more biology, better detection, and we're going to need therapies. And so again, we may talk about that today. The other thing, though, I want to leave you with, and this is really important to remember, is that clonal hematopoiesis is just the sort of canary in the coal mine, using an expression I like to use, because somatic clonal expansion is common across all dividing adult tissues. We now know that age-dependent acquisition of mutations, being every week there's a paper in Science or Nature about a really awesome observation, many of them actually from Cambridge, UK, where they're really, Peter Campbell and the whole group there is really leading the field and charging, delineating somatic expansion across different tissues. And I think it's likely that all dividing tissues have a rate of somatic mutations, and some of these will be a known cancer driver initiators, and that those will be read out because they increase fitness of pre-malignant cells. And this has huge implications in the clinical setting. Diagnostics, a mutation by itself cannot be informative to make a cancer diagnosis. Circulating DNA tests are going to find this in spades. So a cfDNA result is going to be contaminated, if you will, by all of this. And I also think understanding all of this with respect to normal and abnormal physiology in different organs is going to be important in independent malignancy. And you may have seen a paper, I think, that came out of Nature from Chris Walsh that showed that neurons in Alzheimer's disease can acquire somatic mutations that have pathogenic or putative pathogenic roles. But today we're talking about clonal hematopoiesis. And why? Because it's the field where the most awesome science is happening. And that's because first and foremost, the cells are circulating, easier to detect and study. The second is it's a greater capacity to infiltrate different organs. So the pathophysiology in different organismal contexts is much more easy to delineate and to study. And we think it's a vanguard. It's where the great science is. And my colleagues here and many others in the field are really pushing the field. And I think that's exciting, but we're paving the way for all these other tissue contexts. So the way we like to think about it is that you get these early mutations, they increase fitness and expand, and then ultimately in a rare subset of people, you go on to leukemia. But even in the pre-malignant state, you get all these different associated sequelae that we, at least in part, understand or are beginning to understand better and better every single day. And the conundrum I think I want to leave you with, and something that I know that both Kelly and Jyoti and Todd and I have all thought about, is that if you do the math in the United States, there are probably 20 million or more people with clonal hematopoiesis. If you define it with an arbitrary line, and I hope we get to the discussion of whether there is such an arbitrary line, because I think that we have to really figure out definitionally what this means. But if you say detectable clonal hematopoiesis with the standard assay, it's 20 million. But only 30,000 people in the US per year get an AML, MDS, or MPN. So that means that the question is not necessarily why do some people get it, but why don't most people get myeloid malignancy? And we should think about that. But the other thing is that we can begin to trace the evolutionary trajectory from normal to pre-malignant to malignant. And there's different ways to do that. I think Jyoti's work most recently using sort of mathematical models and colony-based sequencing to trace the phylogeny has really illuminated mechanisms of transformation that we really didn't understand before. But the other strategy that we've used, is to use the Mission Bio platform, Lindy and Bobby in my lab, to map out the mutational events as best you can by looking at every cell and piecing together with, again, Markov decision modeling the likely genetic trajectory. And so you can find, the pre-malignant clonal hematopoiesis cells. In this case, it's a DNMT3A mutation. In this case, an IDH mutation. But in some cases, it's a relatively straightforward linear trajectory to AML, but in others, it becomes quite complex and branching, suggesting that leukemia is not an end result of a single clone, but rather a complex ecosystem of clones. And so we think this will teach us a lot about not only why some people get leukemia, but why some don't. And it does suggest that there are key bottlenecks that have to be solved to get leukemia, because the number of mutations that occur to drive and define leukemia are relatively limited. And so what are the critical next questions for the clonal hematopoiesis field? We need to understand mechanistically what the process by which CH is initiated and maintained. We need to understand what these mutations do. I will put out there, and I've spent now like almost 13 or 14 years studying these DTA mutations. We understand very little about how they increase the fitness of stem and progenitor cells. We understand much more about what the subsequent events do to transform cells. What is the pathogenesis transformation to overt malignancy, and how can we therapeutically target CH to reduce the risk of secondary leukemia? And then, of course, are there general lessons to investigate aging-associated malignancies? So a few more comments before I turn it over. The first is that the allele matters, and so does the gene, but importantly, we need to drill down with great granularity to individual mutations. And so this is data on the top from Kelly's work with Ellie Papaemmanuel, looking at the lollipop plot at the allelic spectrum of DNMT3A mutations in clonal hematopoiesis, the very low frequency of 2% or greater. On the bottom is Ellie Papaemmanuel's data with the AML study group from Germany, looking at the same gene in AML. And the point is you get a broader spectrum of mutations in DNMT3A in CH than you do in AML. If we turn our filter up to 10%, you actually get the same distribution in CH as in AML. And this, I think, fits not to give props to Todd's work, but I think he was the first to really delineate this. The idea that there's a low level, high mutational frequency in genes like DNMT3A. So it's not just getting a mutation, it's getting the right mutation in the right organism with the right extrinsic influences that leads for some mutations to pop and expand and most of them not. So it's not mutagenesis only, it's mutations and then expansion we need to understand. And this also suggests we need to understand what every one of these alleles does. And recent work by Peggy Goodell, where she sort of bashed the hell out of DNMT3A, gave us a really great example of how you can saturate to understand what a mutation in a single gene do. And so one strategy we've used is to use the tapestry to link genotype to phenotype, in this case, immunophenotype, because we think if you can look at genotype and immunophenotype, you can begin to ask, what do alleles do? And so this is a patient with two different DNMT3A mutations. The R635Q, in this case, because we have genotype and immunophenotype, is in their T cells. The R882 is in their myeloid cells. So different mutations of the same gene have different lineage output. We think this is going to be incredibly illustrative to ask, what do mutations do in both proteomic and transcriptional space? So multimodal analysis of clone and a mutation-specific phenotypes is going to be incredibly powerful for discovery. The other thing that genotype and immunophenotype is going to be important for is identifying aberrant cell surface marker profiles. And so just an example of a recent experiment that Lindy did, this is an AML patient with all kinds of clones of different genotypes. But the most important thing is right here. This clone, which is the end clone of this patient, TET2 NRAS G12R, has CD14, 123, CD2, 11B60, CD64. And this is an aberrant surface profile you never see on normal cells. So we believe we're going to be able to identify mutant-specific aberrant surface profiles that can be used to diagnose and ultimately to be targeted. And this is going to be the future of these strategies. And we can talk about that. And that may be first play out in leukemia, not in CH. And in the long term, we have to therapeutically target. We need to go from just diagnosing to treating. And we think if we target it, we can make all of these things go away. And so when we think about therapeutic targeting, we need to think about what our goals are. Our primary goal in the early days, including trials that Kelly and Eitan Stein and others are leading, is to prevent leukemia, to intervene to reduce the fitness advantage of clones to prevent leukemia, to intercept transformation. Will we be able to do cardiovascular trials? Maybe. I think the canakinumab trial with the post-hoc analysis suggesting a benefit, maybe, in TET2 mutant patients, maybe suggests there are genotype-specific cardiovascular interventions we can do. And maybe there's other aging-associated sequelae we need to do. But the therapeutic approach, therapeutic index study design is going to be different in each of these contexts. No one size fits all. And I'll leave you with this slide, and then I'll turn it back over to Todd. It's the idea that as we move therapies earlier, we need to think differently about how to use those therapies, what types of therapies are useful, and the therapeutic index that is going to be acceptable. And so we need to think, as we bring in these novel modalities, when we go from AML back to pre-leukemic states, for example, back to, for example, clonal hematopoiesis with aberrant cytopenias to clonal hematopoiesis with normal blood counts, that the bar for what therapies we do and what we're willing to tolerate and what we're looking for will be different. And we need to work on this together. So with that, I will stop sharing and I will turn it over to Todd. I hope that sets the stage for what should be a fun discussion.
Thanks very much, Ross. Yeah, there's so much to go over here. It's hard to know where to start. But since you ended with potential therapeutic interventions, I want to turn it over to Kelly and hear more about what you're thinking in the therapeutic space for clonal hematopoiesis.
Yeah, thanks, Todd and Ross. That was, as usual, really fantastic talk. Yeah, and I mean, Ross mentioned this at the end. I think Ross and I and Eitan have been thinking a lot about, who would be kind of optimal patient subsets to target initially. And, there's several patient populations that we're interested in. Ross mentioned we have a study with Eitan Stein and Ross and me looking at individuals with CCUS, so that's clonal cytopenias of uncertain significance. And this patient population is at very high risk of developing hematologic malignancy. Up to probably over 90% of these individuals will develop hematologic malignancies. So a lot of efforts, including some trials that we're leading, are focusing on that patient population. We have a trial giving IDH1 and IDH2 inhibitors to individuals with IDH1 and IDH2 mutant CCUS. We know whenever you think about interventional studies like this, you always have to, as Ross was alluding to, make sure that the risk-benefit ratio is right. So focusing on these ultra high risk groups, people with CCUS, people with germline predisposition to malignancy, makes a lot of sense. I think it echoes what has been done actually for many, many years now in the solid tumor realm. There's been a lot of success with, colorectal cancer and aspirin, breast cancer and prevention and hormonal therapy. But the other flip side that you also have to keep in mind is, patient uptake, actual real world uptake of these interventions, even if you prove them to be successful, which again, just echoes the fact that you have to make sure that whatever intervention you plan is going to be very acceptable to patients and acceptable in a long-term way, because often for these prevention studies, it takes years for them to show any effect. The last thing maybe I'll leave with is this idea that I think it's pretty obvious trying to intervene in terms of CH and the risk of leukemia, since we know that CH is the first step in this potential multi-step progression. But when we think about intervening on the other health outcomes that CH has been associated with, I think we have to be really careful, because the mechanism linking CH to other health outcomes is clearly not as well established. And so it could end up being a CRP type story where, we think if we lower CH, then it's going to reduce risk of various health outcomes, but it really couldn't be the case that it makes no difference. So with that, I think maybe I'll hopefully spur some additional interesting discussion points. Thanks, Todd.
Yeah, thanks, Kelly. That was great. So just in general, what are your, what do you think are going to be the major therapeutic interventions that come for clonal hematopoiesis? Is it going to be small molecule inhibitors? Is it going to be cell-based therapies? Some combination of all the above? Because a lot of the tools that we're using these days are sort of blunt instruments, and this is a pretty precise lesion. So I'm just curious what you're thinking the future holds for that.
Well, I mean, for certain mutations, it makes a lot of sense, like spliceosome inhibitors, IDH1 and 2 inhibitors. Those are clear, targets for therapeutic approaches that, and for the IDH1 and 2 inhibitors, it has the advantage of being, really well tolerated. So that would be, that's obvious, I guess. It becomes harder for things like for other genes that are actually more common in CCUS or CH in general, like TB53, DNMT3A, like how do you target those kinds of mutations? I think there's the idea that, which is a good idea, like trying to maybe not directly target the clone, but target its ill effects, like canakumumab and TET2 mutant, CCUS. I guess the other, potential avenue for intervention would be to think about how you could modify or protect people from environmental exposures that are known to promote clonal hematopoiesis in specific genes. Like, for example, we know that that smoking seems to be related to ASXL1 mutations, and perhaps probably provides it with a selective advantage compared to other genes. And the more obvious thing would be, chemotherapy and mutations in the DDR pathway, which is something that we're interested in. Nancy Gillis also very interested in. And so, in addition to maybe thinking about risk stratifying people who are about to get solid tumor therapy for their risk of leukemia, and potentially modifying what types of therapy you give, you could think about maybe efforts to try to protect the bone marrow niche for high risk individuals. And those are all things that I think would be really exciting to pursue, and possible, less still targeted, but not clone specific interventions.
Perfect. Yeah, thank you. I think that's a great segue to Jyoti's work. And so, when we think about selection and clonal dynamics as a function of time and growth rates, and so, I'd like to hear from you, Jyoti, kind of what are you thinking as far as, this, I think in many respects, the field has kind of been lumped into clonal hematopoiesis as the same everywhere all the time, which we know is not the reality, and your work is focusing on more nuances of when it's important and how it's important and what to measure and when. So please take it away.
Yeah, the clonal hematopoiesis field is fascinating, and Ross has nicely highlighted some of those early insights that we've almost come around full circle to appreciating, that this idea of non-specific clonal hematopoiesis, we are sort of coming back to that now, because we've taken on the approach of sort of a sort of clonal repetitive whole genome sequencing within single samples to then recreate the history of hematopoiesis in an individual. And that's sort of shown us that clonal hematopoiesis is inevitable by the 7th decade of life, alongside all of our tissues. It's a natural way of human aging. All of our tissues succumb to clonal expansions as we age. And just like, I guess, the rate at which a bank account might grow with a constant rate of interest, the returns are exponential. That is what happens with these clones that are growing over time, with a constant growth rate occurring over a period of years or decades. What you get is exponential returns. And so it appears that you have these small clones emerging that are labeled clonal hematopoiesis as we get older. But actually, the whole of hematopoiesis has become oligoclonal at roughly the same time. So it almost marks this turning point in life where hematopoiesis is at the verge of sort of falling off its perch. And what leads us to get to that tipping point where blood is getting massively oligoclonal? There are just sort of two questions. There's the looking back question and the looking forward. And the looking forward question of what are we going to do about these high-risk clones, because these individuals are at risk of a future potentially preventable malignancy. And that is all the clinical efforts which are incredibly important to manage people who already have these clones. And then there's the looking back, why are these individuals, how did they get to this point? And what germline risk factors do they have? What environmental exposures during their lifetime? And when? We know a bit about what chemotherapy does and bottlenecks, but what about nuances such as immunosuppression or inflammation or obesity or childhood or in utero exposures? What does that do to set your hematopoietic system on a path to these clonal expansions at age 70 or age 80? And that's the kind of angle we're interested in, not just in blood, but blood is, as Ross has pointed out, it's the first system to go to. It's so easy to not easy, but nonetheless, it's a unique system to be able to address these questions in, but we're finding exactly the same thing in other tissues, not just in the context of cancer, but in the context of disease. If there's any selective environment, any insult on a tissue, what happens is the clones that can overcome that insult grow out. So our interest is around those clonal trajectories, and those clonal trajectories, in terms of why do most people not develop any further problems and some people do? Well, what are the trajectories to disease in the individuals that do develop the problems? Is it always that disease occurs on a background of a known driver? Could it occur on a background clonal expansion without a known driver? And at what rate of growth? What is the maximum head start we could have for an acute leukemia? Is it 5 years? Is it 10? What's the maximum head start we could have for chronic myeloid leukemia? Those kinds of things. And I think we also have an interest in trying to understand how different environmental and germline influences that are active throughout life influence that tipping point in hematopoiesis where you become massively oligoclonal throughout your tissue. And the way in which one might get at that is sort of larger cohort studies where you look at clonal heme in the population and try and understand what factors govern it. So our take on it is sort of a evolutionary one, even potentially looking at species other than humans to try and understand, is this how we age? And is this how our tissues age? And is this what's responsible for an aging phenotype? We all know that as we get older, aging hematopoiesis is less able to fight infection and to overcome, the insult of chemotherapy. And why is that? Is that because of massive oligoclonality due to clonal heme, or for some other reason? So that's where our interests are. But yeah, it would be great to see those two ends connect and be able to understand why we get there, to be able to then predict which individuals we could potentially alter those clonal trajectories in. And that's another thing as well. We have this approach of get rid of the mutant cells, but actually maybe in the future we don't need to get rid of the mutant cells, we just need to slow down how quickly they're growing.
Yeah, those are great points. I'm really intrigued by the point you made about this sort of exponential expansion and how early can you intervene. Several years ago, my group was using an error-corrected sequencing approach and found that roughly by the age of 40, you could have some detectable clonal hematopoiesis in the 8 to 10 canonical CHIP genes. We submitted the papers calling it CHIP, and they had a total meltdown because these were mutations at less than 2% allele frequency, so they couldn't be CHIP. So then I thought I was going to be very clever, and I tried to call it microchip. And that got even a more guttural anger from the reviewers. So I abandoned it all. We just called it clonal hematopoiesis. But the idea was that you could detect these things early, and then if you knew there was specific selection pressures coming on, you could intervene. And then to your point, and the work that Ken Walsh and his team and others have done showing that there's end organ damage from some of these clones. Now you think you have clonal variability in an organ that's interacting with clones from the bloodstream that have an altered inflammatory response. And you could see, other things than AML or blood diseases kicking off quite quickly, I think, under those sort of aggregate models. Is that something that your group would be thinking about?
Yes, at the moment we're not looking. Sorry, let me just shut the door one. background noise. At the moment, we are more focusing on trying to understand those different trajectories and the influences on those trajectories. We're not probably the best placed to look at multi-system interactions and multicellular interactions, particularly as we take quite a genomic lens into it. But you're absolutely right, looking at how some of these clonal expansions then go on to interact with other cellular populations, either within the same tissue or across other tissues, is particularly important. At the moment, what we're doing is very much looking back in time, but across a wide range of disorders, to understand the selective pressures on hematopoiesis and their subsequent consequences.
Right. Yeah. Excellent. Yeah. So what do you think, Ross? I mean, how are these, the selection pressure is going to be accounted for? Everybody's mentioned that, I think that a P53 mutation at 0.5% allele frequency may be just as deleterious to the host as an AXXL1 mutation at 5% frequency, but depending on the selection pressures and that the person's going through at that given time, what are we going to do about this?
I mean, I think the hard thing for all of us is that we want to think of these as measuring sort of cancer driver frequencies, and that's not what they're doing. These are measuring sort of the relative fitness of genes that modestly to more than modestly, depending on the gene that you say in slightly, Todd, change the fitness of stem progenitor cells and alter the ability, in the case of some of them, maybe to turn into sort of neutrophils and macrophages that have more inflammatory potential than a wild type cell. And so the way I think of it, and more, is that just like in 1970, we didn't know about HDL and LDL, but it wasn't surprising to anyone that if you ate lard every day, that probably would increase your chance of a heart attack. And then when we learned about HDL and LDL, we began to have an ability to ascertain on the individual basis how someone's both genetics and sort of environment alters their likelihood of getting a cardiac event. This now gives us a look at clones that read out both cell intrinsic and extrinsic factors that increase the risk of things. And we need to treat it like that. And so I think you're right. It can't be just measuring and saying you have a clone of 2%. It's got to be gene or allele specific.
Right. So let me get to some of the questions because they're starting to come in, and anybody on the panel can answer. Please weigh in on your thoughts on this. So the first question from an anonymous attendee asks, based on Ross's point about ctDNA tests, so ctDNA we haven't really talked about, being likely to detect pre-malignant clonal expansion, what are the implications of this for molecular MRD assay development? Do you feel that flow MRD is superior?
I mean, I'll take a stab first because the MRD, as Todd knows, is near and dear to both his and my heart. I'd say we know a couple of things at this point, and I can make a prediction of what I think we're going to learn. We know, first of all, that the presence of residual clonal hematopoiesis is not a priori a harbinger for imminent relapse in patients who've gotten anti-leukemic therapy. Initially, the British Journal paper from the Alliance Ohio State group, and then the really seminal paper by the Dutch group with the Hovan, where they showed that the ability to clear mutations subsequent to DNMT3A TET ASXL1 was predictive of outcome, but not whether you cleared DTA mutations. So we know that. But I think the more important result was buried in that paper. And I think, Todd, you and I were discussing this last week at AACR, which was that if you look at that paper, the Hovan New England Journal paper by Peter Volk and colleagues, they showed that flow-based measurements of aberrant immunophenotypic cells that are residual and the ability to clear or not clear non-DTA mutations had orthogonal value to predicting relapse. And I think what we're going to find is that we're going to have to combine the genomics and aberrant immunophenotype. And I think, for example, in the long term, the ability to do that on a single assay, like, DNA plus protein, or, flow, if you can ultimately flow for mutations, I think we're going to learn things that we'll find that knowing that a mutation is present and knowing whether there is an aberrant immunophenotype are going to together give us the answer. And the future path people that do this will look at that as an integrated report. I don't know what others think.
Yeah, I think the only thing I'd add to that as well is that from a genomic perspective, I think the monitoring aspect of MRD would be key, because if a clone is present at 2%, whether that is growing rapidly versus whether that is static is hugely important in terms of risk of clonal evolution and in terms of the fitness of that clone. And so the monitoring aspect and potentially if flow and genomics could be combined, and then if it could be longitudinal, then one can start to really predict future trajectories. And I think that is where the risk stratification might come in the future.
Yeah, I would add, there's been no bulk NGS assay for MRD that's really outperformed flow. And, as such, a lot of people do them, in addition to flow, or, I don't know that anybody does them in lieu of flow, but, these bulk assays, to both of your points, have not been able to capture. It's either the surface immunophenotype that seems to be really important, or the point mutations or the genetic lesions. And to the data that you showed, Ross, it seemed like there's definitely value to combining both of these to really gain granularity in what's there and what's potentially relevant. Has anybody seen anything for ctDNA with clonal hematopoiesis? I know people have dabbled in that. I haven't seen much that's been. Anybody?
Our internal assay, MSK Access, uses paired cfDNA and blood to filter it out, and that I think may be a little bit different than what a lot of the commercial providers are doing, and we think that that's important, and we're hoping to generate enough data to figure out whether that's instructive. I haven't seen the full data set. I don't know what others think.
Yeah, I know there's a lot of concern. Anytime you talk about a ctDNA assay for a solid malignancy, there's always concern that the mutations you're going to pick up in the ctDNA are actually clonal hematopoietic mutations, but I don't know of anybody using them to actually do surveillance on clonal hematopoiesis.
Yeah, I don't know.
Yeah. All right. Questions are rolling in here. So the next question is from Rose Beck. And Rose asks, where is the role of the master regulator concept, i.e. that RNA expression signatures might be most important for parsing phenotype, not just individual mutations, which I think is relevant to our last discussion.
This is what I keep trying to tell grad students joining my lab is the next thing to figure out, which is, can we identify core transcriptional circuitry that's associated for driving these things and figure out how much of that is, engendered by acquisition of the mutation, or by the mutation acquired and a cascade of subsequent epigenetic remodeling and a subsequent transcriptional signature, or through independent stochastic transcriptional changes. You know, the observation that you get mutations at 0.01% and only a fraction go to 2% and only a fraction of those go to 20% suggests that just having the mutation itself is not enough. And the question would be, how much is the mutation setting off a cascade that is still driven by the mutation, or does the mutation need other things to happen that are independent, is a really interesting question. And then what the biology is all that, I think that needs, but I think that's where the basic science is going to go.
Yeah, Kelly or Jyoti, have you guys worked on any of the RNA transcriptional signatures for these?
I haven't. I think it's a great idea. It's about you know, just you have to have the right you know, samples to be able to do that clearly. But we know from other, you know, from hematologic malignancies and solid malignancies that the combination of mutational features superimposed on transcriptomic profile adds additional prognostic value. So it would be really cool to look at that as in terms of relation to maybe CH and risk of leukemia, or risk of you know, clonal expansion maybe, but we haven't done specific work on that. But again, it's definitely something that should be done in the future.
I think also with clonal heme, what's emerging now is there's so many clonal expansions that do not yet have a known driver. And so what is driving their clonal expansion? Is it at the transcription level? Is it at some other epigenetic level? I think the challenge with studying RNA expression is, you know, at what point in hematopoiesis is this universal signal of selection active? Is it just at a stem cell level, in which case it's pretty challenging to study using the approaches that we have, where we're looking back in time and we don't quite know the phenotypic or the cell of origin in which these drivers occurred. But certainly that's, as Ross has pointed out, that's where some interesting biology will lie. You know, these mutations are occurring in many different cells and many different individuals, and they don't often have the same consequence. And is that because of a cell of origin and because of particular, you know, fertile ground of epigenetic plasticity in some way that allows these mutations to then have their effect?
So has there been any work with the Wellcome Trust on the epigenetics or methylation or surrounding these questions?
We're dipping our toes in methylation at the moment because it's a little bit more stable as marks in terms of the cells can change, but some parts of the methylome don't. We have dipped our toes in transcription, but we find that we're picking up basically the transcriptional state of the cells that you've sequenced, which is expected and not really telling you of how they've been altered historically when the mutations arose.
Right. Okay. Great.
But you need a stem cell pure population to sort of try and address those things.
Yeah, there's so many things that can drive these gene expression profiles and toggle them a little bit this way and a little bit that way. I've always had trouble when you see them. How do I know that, you know, this 10% higher expression is relevant to, you know, not that it's not important, it's just very, very difficult to account for. But, you know, hopefully we'll get there.
And Todd, I'll just add, there was, I mean, one really cool paper related to this was from Sid Jaiswal's group. They looked at, you know, epigenetic markers of epigenetic clocks and how maybe CHIP, in addition to methylation-based markers of aging acceleration, might predict, I think, survival. And they showed that the combination of age acceleration plus CHIP was even more prognostic. So there definitely is something that there's going to be a lot of information that we get from these multi-omic assays for sure. And we're just at the tip of understanding.
Yeah, thank you. Yeah, that's a cool paper. I know that paper.
Yeah.
All right, Mary Donahoe asks, the numbers disparity between clonal hematopoiesis of 20 million in the US per year versus leukemia or other blood illness of 30,000 per year suggests that a strong repressive or surveillance mechanism gets depressed. Is there any data for this? I think, you know, that's sort of the million-dollar question, which is what Ross posed in the beginning. It's why aren't more people getting leukemia? So what is that mechanism that, you know, it's easy for me to say, well, the immune system, you know, is doing surveillance and sort of takes out these bad actors, but it's not foolproof. There's some bleed through, whatever problems arise. But yeah, go ahead.
But it's not, right? I mean, doesn't look like in the 30 million that the clones dissipate. I'm not aware of a lot of data that has shown that there's dropout of these clones. I'm sure there will be dropout, but if you look at the longitudinal data with the caveat that it's in limit short follow-up, and Kelly's done it, it's not like we saw in the serial samples, and these are even in people off therapy. So like we've taken away, it's not like clones drop off. They just kind of hang around. I think the immune system could be important, but as you say, Todd, it's a hand-waving explanation we all use.
Yeah.
I think the observation to me, I don't know what the others think, that despite having these early events that we know can increase stem cell fitness and frequency, that they don't go on to develop 1,000 different mutational sort of journeys to leukemia, but like 15, right? I mean, NPM1 here, CBLP alpha there, P53 complex karyotype. It's a pretty limited number of paths. And that does suggest potentially that there's something to the very small number of bottlenecks that have to be solved, and a limited number of genomic solutions to them. And very different, maybe the mutational rate of the tissue is important too, like versus melanoma that's unexposed. But, you know, it's really illustrative to me that TET2 mutant overt disease has a co-mutational pattern of only 20 different genes, right? And what that means is the rest of the genome can't be the solution. And maybe that is part of it. Although I agree that the extrinsic factors, and this is something we do a crappy job of modeling in mice, because they're in these germ-free facilities, that, you know, we have to also acknowledge that the outbred human and the environment they're in is probably a huge arbiter of that too. And that's very hard to study mechanistically.
I think also that the stats there in the Q&A, the 20 million versus 30K, I think that's also testament to the fact that clonal heme is not sufficient to cause AML. It's not sufficient on two levels. It doesn't have the right mutations, and it doesn't have the right growth rates, and it doesn't have the right clonal evolutionary trajectory. And so we all have clonal heme, not just 20 million per year. You know, for our data suggests that anyone over the age of 70 to 80, their entire over 50% of their hematopoiesis is clonally derived. But that is just normal aging. That is not, clonality does not equate to cancer. And there, mathematically, it's very unlikely to then get the right number of events and growing at the right rate to then present with a cancer phenotype within your lifetime. And clearly, evolutionary, we've selected for a hematopoiesis system that relegates most cancers towards the end of life, and we've successfully managed to do that evolutionarily. And so what we're getting is the maths playing out at an older age, because there will be a fraction of people that have the right growth rates and the right combination of mutations to then present with a disease phenotype.
Yeah. And when I, mentioned the immune system, and again, I have no data to support this, but I think when we track and say, well, those clones don't disappear, so the immune system isn't really doing anything, but you're measuring a single mutation. And it's, to your original example, Ross, you showed, there was this secondary JAK mutation, and that's where things took off in this particular patient, so maybe those multi-hit clones are the ones that do get removed, or maybe there's not the right selection pressure, but yeah, that's a major question for the field that needs to be addressed. So good question. All right, here's another one. So an anonymous attendee says, thank you for the great talk and for very comprehensive information. Is physiologic cellular aging, and in parentheses, they say, I am thinking about in utero specified systems like telomere or telomerase axis, Are these coming first or after acquiring of physiological hematopoiesis? Is physiological cellular aging?
I don't know. Didn't
after-
Jyoti, didn't you look at some of the telomere aspects in your recent paper? Did I have that right?
Yeah. I'm not sure I fully understand the question, but I think certainly we have two things that we've learned from looking at both telomeres and looking at across tissues in humans when we kind of use this phylogenetic approach across multiple tissues to go back in time, is that all of our tissues share the same original cells. Now, that might sound obvious, but what's not happening in fetal development is that you have a set of cells that are earmarked for liver, and a set of cells that are earmarked for blood, and a set of cells that are earmarked for muscle. What's happening is every cell is derived from the same set of ancestral cells that equally contribute to multiple tissues. So in that respect, I think physiological hematopoiesis comes early and is derived from a wide selection of cells that are present within the developing embryo. What we found in terms of the telomere data within these individual cells, essentially from these individuals with a myeloproliferative neoplasm, and indeed in normal hematopoiesis aging, although that much of the telomere data from that paper isn't in the paper at the moment, is that the telomere measurements that we're making seem to be a reflection of the cell division history that the cell has clocked up over its lineage. And so the telomeres were shorter in tumor cells, but purely, we think, because those tumor cells have racked up more cell divisions over their history of their ancestors. And so for me, the telomeres seem to be consequence as opposed to cause. But at the same time, there are all these clonal expansions occurring within blood that do not have known driver mutations. And maybe how short or long your telomeres are is an input into whether a cell can expand upon acquisition of a driver mutation. There's some interesting papers, Pradeep Natarajan's got a lovely paper show that the telomere length is actually causative using Mendelian randomization approaches, so that if you have longer telomeres, you actually potentially have more capacity to expand if you get a driver mutation. So maybe there is a sort of bidirectional effect with telomeres. If you are expanding, you will shorten your telomeres. But if you have longer telomeres, you have more fuel to expand to begin with. And that might influence on the fitness of a driver in a cell, as opposed to whether it might then sort of burn out quickly. So I think there's a lot to learn about how telomere measures feed in to clonal expansion, as well as a result of clonal expansion.
It's cool biology, yeah. Interesting. All right, so here's a question I'll give to you, Kelly. I think you might be the right person for this. Jessica Galen asks, if you have multiple clones with actionable mutation profiles, how do you decide on a targeted treatment regimen? Are you studying combinations of targeted inhibitors based on clonality?
That's a great question, Jessica, thank you. And the easy answer is that we don't have a lot of options. So it becomes very easy. So, if someone has an IDH2 mutation and a DNA3A mutation, then all we have is an IDH2 inhibitor. So that's the short answer, but it's a great question. I mean, more philosophically, now than practically, but in our trial, a big safety concern is very reasonably what happens if you inhibit an IDH2 mutation and the clone bearing that mutation. Like, could there be, by doing that, could you be altering the clonal trajectory of other clones that bear other mutations that and you kind of enable them to expand? You kind of facilitate their fitness in some way or alter it. And so as part of any clinical trial where you're doing things like that, you clearly need to have surveillance closely for the expansion of other mutations. And we have, for example, in the trial written out that information will be used in the context of other clinical signs to monitor for possible progression of disease being led by expanding other clones. So yeah, it's a big concern, and nobody really knows what's going to happen. The natural history of this. But yeah, and I think, as we develop more options clearly for additional inhibitors that could be used in this setting, or layering on additional targeted therapies, that would make a lot of sense. And is essentially kind of already done in some ways, for other diseases, not in the preventative setting, but in advanced malignancies.
Great. Any other comments? That was great. Thank you, Kelly. So Yang Li asks a question and says, it appears that AML and MDS are dominating the discussions on clonal hematopoiesis. How about the roles of clonal hematopoiesis in the risk for lymphoid cancers such as ALL, CLL, Hodgkin's, non-Hodgkin's, and multiple myeloma? Any thoughts there?
I can add something. I don't know if Jyoti or Ross, maybe they probably also have great insights, but maybe just to mention that when we're talking about clonal hematopoiesis, it can be driven just like different types of mutational events lead to cancer, like structural events, copy number events, point mutations, indels. The same thing happens in clonal hematopoiesis. So we know that large scale and smaller scale copy number events can lead to clonal hematopoiesis. And a lot of that work has pretty powerfully shown that copy number events are particularly prognostic for lymphoid malignancies like CLL especially. Actually, the prognostication that you get for CLL, looking at purely copy number alterations, is better than you get for myeloid malignancies. So definitely there's prognostic value in tracking individuals for both myeloid and lymphoid malignancies, but the types of mutations that are prognostic differ. And Ben Ebert also has work on this, comparing myeloid and lymphoid drivers and how that and the prognostic effect of those in determining myeloid versus lymphoid malignancies. So yes, it's relevant when oftentimes we kind of forget that CHIP isn't just SNVs and indels. And so I think that'll be a really interesting field, especially now as more and more people are able to combine data on copy number events and SNVs and indels together. We're understanding how they layer together to promote both lymphoid and myeloid malignancies.
Good. So, yeah, well, the questions are pouring in. Here's one from an anonymous attendee. So this is a little bit long, but I'll go ahead and read it here. Do we have a good idea of how many of the myeloid gene variants may be coming along as constitutional? Do we have data on young kids for all of the common CHIP gene variants? If some are constitutional, do we know how that affects the patients when a true myeloid neoplasm arises, similar to the TP53 scenario? This is a challenge when we see variants such as TET2 variants are classic for this issue in a patient's tumor, and see it also at a relatively high percentage of normal patient databases where it's been suggested that it is of no consequence due to this. My feeling is that one can certainly not conclude this, and that it may well be biologically relevant once other variants arise, even if it was there from birth or acquired as CHIP. Thoughts? So I can start here. So I've got the good fortune of collaborating with Jamie Blundell, and Kelly has recently joined us in a project that's similar to this question, I think, where we're looking at samples from birth, childhood, adolescence, and young adulthood in the same individuals. And so I'm a pediatric oncologist. I've always wondered, are there different genes? I see a lot of pediatric malignancies as defects in development rather than defects in normal cellular maintenance. You know, that's a very generalized notion, but so we've done a lot of the error-corrected sequencing, including genes that are important for pediatric leukemias versus the canonical CHIP genes. And this is all preliminary. It's not published yet. We're still working on it. And Kelly may add more to this. But what we're seeing is that there is clonal hematopoiesis at birth and in childhood. It's roughly 20 to 30% of the patients that we've queried so far. But it's not in the canonical CHIP genes. These are in different genes than what you see. And then for our data, where we have a limit of detection of 1 in 10,000, around age 40, there's a very, very sharp inflection point where all of a sudden the DNMT3As and the TET2s and the AXXL1s, they're just there from there on out. So, you know, what I was wondering was, are some of these clones being transferred from the maternal circulation to the fetus? I don't see much evidence for that in our data anyway, but I'm sure it's possible. But to the question asked by the attendee, you know, we're just not seeing that the children have very rare clonal mutations in DNMT3A or AXXL1 or TP53. You know, similar to later in adulthood. That's what we've got so far. Kelly, you want to add anything to that? Any thoughts?
Oh, flip, I'm on mute. No, I mean, Todd, I think there's, you know, this finding of yours, this preliminary data, makes a lot of sense when you think about other types of cancer. Like, when you think of cancer in general, right? And so, say we look at someone that has a germline predisposition to whatever. You see cardinal cancers that happen in young people, and the cancer risk just stops when they reach a certain age. And then, as opposed to, you know, you see some cancers that they just always occur right in older age, which reflects that there must be some type of advantage to certain clones depending on the age of the individual. And I guess also getting to, maybe there was a point that was almost a little more technical here. I think that they were trying to make that about distinguishing constitutional clones from constitutional mutations or variations from CH. This is very challenging. And I think, you know, one of the ways it's clearly, I mean, the way of getting around that is to do this types of testing using matched tissue, which we know, you know, from work with Ross and me using the impact data and other data sets using matched tissue, that really helps you parse out constitutional variants from true clonal hematopoiesis. And I would say it also has taught us that the kind of the bioinformatic filtering that we can do to parse those things out actually works quite well. And so if you're careful enough with your filtering, I think it is possible basically to discern germline events from clonal hematopoiesis, even in a young individual where clonal hematopoiesis is quite rare.
One of the most important things we do in our clonal hematopoiesis clinic is do matched blood non-hematopoietic sequencing to sort of settle the score in people that come to us when the referring doctor and patient have no idea which it is. And we find it many times, it's incredibly clarifying. And the other point I'd make is that there have been a number of kindreds, Rhotic Skoda, I think, as well, Pro as well, where TET2 germline variants have been observed and where they, at least in family pedigrees, seem to increase the propensity for myeloid malignancies. So there's absolutely no doubt that those do occur, but the data would suggest to date that those are exceedingly rare compared to somatic variations in the same genes, the same positions. And so although it's an important thing, the critical thing is to delineate, I would argue, who has somatic variants. And it gets back to Jyoti's point that once you know it's somatic, you can follow it over time and ask whether it's changing, which may be the most important thing you can actually help to risk stratify a patient.
Great. Okay. So here's a question from Jatinder Lamba. Asks, most new data is showing less than a 15% correlation between transcriptomics and proteomics. I would love to know the thoughts of the panelists on this issue. Who wants to dive into that?
I mean, that's like a sort of existential biological question that, you know, we can spend hours talking about. I think we always have to remember a couple of things. The first is that evolving, and that RNA-seq, as good as it is, whether it's single cell or bulk, is measuring the abundance of an mRNA at that moment, not its production, nor its decay, nor the dynamic changes. And it may be that those aspects are more important to what gets transcribed than just the absolute measurement at that moment. The second is that our ability to enumerate proteins is in like the early days. And so, probably the most important and neat thing is, if you follow the field, is that the sort of biology of RNA modifications and things that affect even RNA processing, even changing bases in RNA, something that I've been almost like as a DNA guy saying, oh, come on, it's all in the DNA. But there's more and more data that some of these RNA editing are really important in pathogenic processes. So we just have to keep an open mind and say, this is what the data shows, and acknowledge that there'll be additional aspects, and always ask with these emerging technologies how they can be adapted to measure the additional things that we're beginning to learn, and not just rest on what they already can do.
Sounds good. Thank you. All right, Jessica Galen has a question for you, Jyoti. So Dr. Nangalia, have you looked at clonal hematopoiesis in populations with known environmental exposures to carcinogens, such as those living near radioactive Superfund sites?
I think that would be a great experiment to do. We haven't so far. I think we're, you know, as a group at the Sanger, there's a lot of work on skin and other tissues in the context of carcinogens, not yet on blood. Blood seems to be quite beautifully protected as a system. Maybe evolutionarily we've required that, apart from the chemotherapy setting. So there is a huge amount of new signatures emerging in response to drugs that we use quite frequently. I think the most interesting one that emerged recently was from the Van Boxtel group, when they found, you know, ganciclovir-induced mutagenesis in stem cells. Specifically in terms of environmental exposures, no, not yet. If I was to do that, it would be an exciting experiment. I probably wouldn't just look for clonal heme. I'd want to look at mutational processes. And in order to look at mutational processes, you then have to rely on there either being a clonal expansion, or you have to go in at the single cell level, or you know, potentially things like NanoSeq, where you can really harvest single cells almost. You can use a bulk sample to get single cell insights. So that would be a great experiment to do, but one would want to look at the nature of the DNA damage and then what sort of driving mutations are coming along in that context.
Great, thank you. Priya Chakalingam asks this question: high-risk TP53 clonal mutations in sickle cell disease patients present before an allo-hematopoietic stem cell transplant and then expand after transplant and contribute to leukemia risk. Is there value in preserving mononuclear cells for single cell analysis in addition to banking peripheral DNA from myeloid gene panels? Provocative question.
Yes.
Yeah. Yeah, of course, right? I agree. I think, outside of the sickle cell world, we've published some work in patients that receive bone marrow transplants for, different hematopoietic malignancies. And we found that the clonal hematopoiesis mutations in the donor. So, and Jyoti made this point early on, you've got a donor who's healthy but has whatever physiologic clonal hematopoiesis, giving their bone marrow to somebody who's had immunosuppression and antibiotics and potentially radiation and chemotherapy and all manner of selective events. And 100% of the clonal mutations that we could track with our technology engrafted in the recipients. And, we had follow-up a year later. A lot of those clones had tapered off, but some had actually persisted and expanded. So those weren't sickle cell disease patients, but I do think that there is going to be value in characterizing what a bone marrow transplant recipient is going to receive, and then monitoring that post-transplant. I think that'll be fascinating. Any other thoughts, Kelly, Jyoti?
I think the value of young blood is going to become more and more apparent over time, just having a highly diverse population of stem cells, to the point where it does end up being a philosophical discussion. Are we all going to be wanting to bank our young blood for an autologous transplant when we're older? The clones that are being deposited becomes more and more important as we learn about how they will behave in a different environment.
Absolutely, yeah. So Tom Conklin asks, how will aberrant protein expression affect our therapeutic strategies for both CAR T and BiTEs?
I can take a crack at it. I mean, it's my opinion that we've done a really crappy job in the field of measuring the expression of antigens or combinations of antigens on clonal aberrant bona fide cancer cells compared to cells that are not. And the problem in those fields has been a paucity of high quality target data and actual primary cells that are proteins on the surface versus transcriptional data or predictions. And it's my hope that as we generate this data as a field, where we really can show that a particular antigen or combination are specific to clones that aren't present on wild type cells, that it will maybe give more precision to these incredible modalities that are being developed.
Any other comments? All right, thank you, Ross. So Babal Jha has a question for you, Ross. What is the basis for such a long latency for the chip clone? I think this is in reference to the one that you showed in your opening slides. The preclinical model, particularly for TET2, is so quick.
Well, I'll say two things. The first is that I think the model isn't that quick. And I just want to take you through the model that in a TET2 knockout mouse, where we engineer either all or in a mixed experiment, 50% of the cells that have the mutation. So you've stacked the deck, right? Half the stem cells already have TET2. It still can take 16 to 32 weeks in a mouse, which is years in human time for to overpopulate, and 2 years in a mouse, which is a lifetime in a human to develop overt malignancy in a subset. And as we've moved from MX inflammation-induced models, for example, to SCL-CRE, we see many of the mice never get leukemia. So I would actually quibble with the idea that the models are quick. I actually think the models are slow. I'll just remind you, though, that models where you engineer the mutations in every cell, you've already done the job for clonal hematopoiesis with your inducer, and the mutations actually, its job has been done for it. You've already expanded it, right? So whereas doing the right experiment in a mouse would be to engineer TET2 in a very small number of cells and follow them over long periods of time and see how often they develop malignancy. That experiment's not been done with precision. But the converse I will take you is something that I've struggled with in our data and in Jyoti's data, which is, how in the hell can somebody have, fine, take DNMT3A TET ASXL1, but how can someone have a JAK2 mutation for 10, 20, 30, 40 years, and then later they get disease when JAK2 in a mouse model, I think, has a much faster penetrance? And I think it's an interesting enigma that we need to understand. I don't know what Jyoti would think. I think it's a fascinating observations of the human that are not fully explained by the mechanistic studies that we have to date.
Yeah, I think JAK2 is so incredibly common in the human population. I mean, the Danish data suggests that 1% of the population has a JAK2 V617F of 1% or more, which would suggest that the level of individuals with a VAF of under 1%, I mean, maybe most of us have a very, a mutant HSC with a JAK2 mutation. I suspect it's a very weak driver, and it just doesn't have a strong clonal advantage in the setting of the other HSCs. How that's different in the mouse, in the context of having a single mutant HSC appearing in a mouse setting, I'm not sure that that experiment has actually been done in the context of native hematopoiesis. So not sticking it into an irradiated mouse that's very, very sick and some competitive HSCs and seeing which one grows out in an inflammatory environment, but actually in a really healthy mouse, making one stem cell mutant and seeing what happens, potentially not very much. We don't know. So I think it's a really weak driver, and the patients that develop MPN for some reason have some propensity to select on it, but I might be completely wrong.
I think that experiment has to be done, and one of the things we believe is that in mice you have to do inefficient mutational acquisition in a sort of unconditioned host and develop and I actually think, and I've been begging my lab and the folks at Mission Lab before talking there to develop the mouse tools to measure these things in preclinical systems, because if you had single cell tools to measure it, you want to marry the inefficient activation to, you got to track in the mouse. And I think we're going to need the same tools in preclinical models we have in the human to do exactly the experiment you described, Jyoti.
And I think the results of that experiment will be negative, and so no one will do it, but it's the most important negative.
Oh, we're going to do it. Don't worry.
It's the most important negative result to show that
don't worry, we'll do it. I promise to you and everyone on the webinar.
Thanks, Ross.
And I guess maybe the other thing would be to think about the germline genetic background, right? I mean, we know clearly from work from Ross and Jyoti that you're better to talk about it than I am, but that, germline genetic factors make a big difference in who develops MPN. So, somehow kind of recapitulating that would be important as well.
And I think more so than other diseases, because it's such a phenotypic disease. So we've got some work with Nicole Soranzo and UK Biobank, where we're just trying to look to see what germline influences, and even your germline influence of where your hemoglobin and your platelet count and your white cell count is going to sit has a strong influence of the clonal fractions of JAK2. They influence whether you select on a driver mutation, and they influence what kind of classification of disease you're going to get. So I think the point you make about the germline is particularly important for some hematological diseases, perhaps not, spliceosome driven ones or anything like that, but for blood count parameter changing diseases like a JAK2 mutation may be more important.
Yeah, here's a related question from Shivani Golem. And they ask, from your experience in people whom you've identified clonal hematopoietic genes, I think they probably mean mutations, what was the time duration for this clonal hematopoiesis evolved to other gene mutations? I think what they're asking is, sort of what's the average amount of time where you go from mutation 1, add a mutation, and now a problem occurs. I think that's my interpretation. And I think that's highly variable, right? That's what we've been talking about. It depends on selection pressure. It depends on, what is the mutation, the first mutation? What is the second mutation? Is there any germline variability there? What's the epigenetic state? I mean, there's a whole list of things that go into that. But I think from the work that we've done in a limited number of patients, because these phylogenetic studies, the caveat is there's like half a dozen to a dozen patients. It's generally at least a decade between the second driver, and that might just be maths. What are the chances of, if you were getting 20 random mutations within our 3 billion bases per year in a stem cell, it will take us a while to randomly, by chance, acquire a mutation in a gene that will have a consequence.
So we're finding in the context of both clonal heme and MPNs, which are perhaps the slowest end of the spectrum, that it is around, 10 years to 20 years till you get the second hit. What we don't know is how that would change if the clone was growing really, really quickly, as might occur in conditions that we haven't studied yet and that are actually quite difficult to study, like the leukemias. What would happen in that context? Interestingly, in something like chronic myeloid leukemia, which you typically believe to be quite an explosive sudden onset disease, NGS and additional driver mutations just don't seem to have a massive role clinically because they're just not universally present in everyone, probably because the disease is just too short-lived in terms of its history to have acquired. So maybe there is a minimum amount that you require for a second hit to be mathematically plausible and for it to grow out. But so far, we've only detected ones that take a long time to get the second and third hit.
Yeah, that's great. So let me add to that a question from Edward Evans. So Edward asks, given all the factors being juggled in clonal hematopoiesis to malignancies, treatment, and prevention, is there any work being done on cell states? Are we as a clonal hematopoiesis community piecing together cell states with all of the various omics?
Yes. I mean, I think the point is that the DNA plus protein is beginning to link clones to immunophenotypic states, if you will, and as we begin to be able to do RNA as well. The challenge will be that the cell states that occur in normal hematopoiesis may be sort of disrupted or become a caricature in the malignant setting. So we have to be sort of open-minded about what our definition of a state is, and yet try to use the framework of hematopoiesis so that at least we have somewhere to start. So, to sort of like layer it. And I think people, Paresh Vyas and others have done this previously with flow-based sorting strategies followed by mutational analysis, and John Dick and others. But I think we're going to learn a lot, and we're going to redefine and rewrite these rules. And the thing here is that in general, the problem has been the field has done a beautiful job with single cell RNA-seq of defining cell states, but that's mostly done in the normal. And we're never going to be able to really understand how it's important to cancer until you add clonal information to that. And that's where you need a DNA out vision of this. Whereas I think ultimately, the challenge has been that, when you look at, these beautiful work done in prostate and lung and all these other things, single cell RNA-seq in the normal, and people find all these different basal and luminal and whatnot. But then you do the prostate tumor cells. If you don't know what cells have the mutation, you can't then assign the clonal composition of those cell states. And we're going to need all that information together in the long term.
Great. Okay. So an anonymous attendee asks, how do you follow up on chips that are pathogenic or likely pathogenic versus the variants of unknown significance? It would be crass for me to say do a lot of mission bio testing, but I could throw that out there. But no, I think we've sort of hit on that, right? There's not going to be a single mutation that's going to drive this, so you're going to have to follow up and look for additional mutations to come along. What do you think, Ross, in your data, as far as adding the surface immunophenotypes to that? Does that help sort of tease apart pathogenic versus VUS, or is it just simply the DNA, the gene, the mutation?
I think that's a great question that should be studied. We've not thought about it. Actually, it's a really good one. That the question that you bring up. I think what historically we've struggled with is that the way we ultimately figure out what is relevant to disease is either by showing that it has some prognostic value, some therapeutic value, or by doing really, going back to the lab and showing that it disrupts the function of protein. And any of those can be sort of like, it's like in a court of law. Those are the abundance of evidence we use to make a case, right? But no one of them is ultimately definitive unless you have a drug that targets that mutation only, makes it go away and the patient's better. Everything else is sort of is and I think what you're making the point is that if we could show that mutations or we show that variations are associated with aberrancies in the immunophenotype, that might be a really nice direct, readout of what that's doing. And if we could do that systematically, for example, for DNMT3 and type 2, that could be incredibly illustrative, in my opinion. I think it's a great point, Todd.
I think the thing I would add to that is, I guess it depends on how you define pathogenic. Is it that it's going to lead to organ dysfunction? Is it that it's going to lead to an increased risk of something in the future? Or is it that it's just under selection? One could argue that if it's growing and it's under selection, then it's clearly providing a fitness advantage over some sort of, competitor stem cells or environmental insult that's being experienced. And maybe if a clone is growing, then it is one that requires monitoring and follow-up, because it's the one that's most likely to cause problems or potentially clonally evolve.
Thank you. So here's a topic we haven't really touched on, which is metabolomics, right? So an anonymous attendee asks, cholesterol is essential for proliferation. How could this affect clonal hematopoiesis? Is there any rationale to consider this? I don't know if it's just cholesterol or more the metabolomic state. I don't know. Has anybody done any work on that? I have no idea.
Best work done is by Alan Tall and colleagues who've shown that manipulating lipid states synergize with these alterations to promote atherosclerosis, and an earlier work showing that it can change hematopoietic stem cell fitness. But I think there's like a mountain to do there. Sorry, Jyoti, I cut you off.
No, that's, yeah, I was going to mention data from the liver. So there's a recent paper, Peter and Stan Gar is the first author, where they've looked at chronic liver disease that's riddled with clonal expansions from cells that are shirking their metabolic duties by overcoming the way in which they metabolize fats. And what's happening in that situation is there's a metabolic insult, and it just provides a unique selective environment whereby cells that can overcome that insult grow out. So the next question is, what other tissues does that affect? And is it some sort of pan-body clonal dynamic change and shift, or is it disease specific? And I suspect there will be insults that affect all tissues and insults that are tissue specific. But it would be a great thing to study.
Yeah, that's great. So here's a topic I've heard a little bit about in the last couple of weeks, which I'd like to get your thoughts on. So clonal hematopoietic cells in a solid tumor, right? So if you could characterize the CH profile in a solid tumor versus the peripheral blood, is that going to look different? And then if I radiate that tumor, am I going to create clones or cause clones that have an advantage to proliferate that then have some external effect? I've been asked this question 3 times in the last 2 weeks, and I never even thought about it before, but it's pretty cool. Anybody with any thoughts, or is that just...
I mean, we did some work with Larry Norton and Elizabeth Coman showing that in a small number of women with breast cancer, that the variant allele frequencies of the mutations were higher in the infiltrating tumor than in the circulation, suggesting that there is some tropism to the tumor site. And we have a lot of data in the lab in preclinical models now asking whether mutant hematopoietic cells can infiltrate tumors. Our gestalt is they affect more the response to therapy and proclivity for dissemination than the ability for solid tumors to initiate and grow in their primary site, but that's what my early inclination suggests. The other question you're asking, Todd, which I think I want to broaden out, was what the hell is radiation doing in all this? In all of the studies, including Kelly and study, which was our first, and then Kelly and Amit's, radiation's a bigger fitness changer than chemotherapy, which was the opposite of what at least I thought when we began this work. And the question is, what's radiation doing? Getting back to Jyoti's point, almost every model everyone used to study hematopoietic stem cell fitness requires a radiation as a conditioner. And so you can't retoast a mouse after you've already toasted it. And so I think we need to move to more elegant systems where irradiation is not used to establish the model, and then use radiation as a perturbation, both locally and systemic and whole body, but mostly and ask, is the effect due to the radiation or due to the a response of the organism to the radiation or some combination thereof. And I think there's intriguing ideas you can come up with all that, but that's like unexplored and desperately needed, in my opinion.
Great. Thank you. Well, we got 2 minutes. So we've gotten through almost all the questions. Any last thoughts, comments, suggestions, anything from our panel, or any other questions that need to come in from the audience? Because I think this has been a really exciting discussion. I've had a lot of fun just talking science and throwing out ideas. So yeah, if there's nothing else, last chance, gone. So I'll go ahead and wrap things up. So I want to thank all of our attendees for taking time today to join us. I certainly want to thank our panel, Ross and Kelly and Jyoti, for taking their time and lending their expertise. And so on behalf of Mission Bio and everyone there, thank you again, and wish everyone a safe and healthy weekend. Thank you.
Thank you, Todd. Thanks, Ross and Jyoti.
Thanks, Jyoti, Kelly, Todd.
Nice to see
Be well, everyone.
Thank you.